Good morning, this is Dr. Will, CBMD, PhD. It is Thursday, September 8th, 2011, 12:04 a.m. Eastern Time. Quick note. Mammal cells usually contain two sets of chromosomes, one set inherited from the mother, one from the father. The genetic information contained in these chromosome sets help determine how our bodies develop. Changes in this genetic code can lead to or increase the risk of developing disease. To understand how our genes function, scientists manipulate the genes in animal models, such as fruit flies, zebrafish, and mice, and observe the effects of these changes. However, as each cell contains two copies of each chromosome, determining the link between a genetic change and its physical effect, or a phenotype, is immensely complex. Good morning, this is Dr. Will, CBMD, PhD. It is Thursday, September 8, 2011, 12:02 a.m. Eastern Time. Quick note. Scientists create mammalian cells with single chromosome set. Science Daily, September 7, 2011. Researchers have created mammalian cells containing a single set of chromosomes for the first time in research funded by the Wellcome Trust and EMBO. The technique should allow scientists to better establish the relationships between genes and their function. Good afternoon, this is Dr. Will, CBMD, PhD. This is Wednesday, September 7, 2011, 12:18 p.m. Eastern Time. Quick note. Growing meat in the lab, scientists initiate action plan to advance cultured meat. Science Daily, September 6, 2011. Late last week, an international group of scientists took a step closer to their goal to produce cultured meat. They agreed on important common positions about how to bring the research forward during a workshop in Göteborg, Sweden, arranged by Chalmers University of Technology and European Science Foundation. Good afternoon, this is Dr. Will, CBMD, PhD. This is Wednesday, September 7, 2011, 12:19 p.m. Eastern Time. Quick note. Many technology components are now coming into place in order to realize the concept of cultured meat. This includes a cell source that is possible to use, several alternative processes to turn these cells into muscle cells for meat, and nutrients free of animal components which can be produced from sunlight and carbon dioxide. Good morning, this is Dr. Will, CBMD, PhD. It's Thursday, September 8th, 2011, 12:06 a.m. Eastern Time. Quick note. Evidence for persistently iron-rich ocean changes views on Earth's early history. Science Daily, September 7, 2011. Over the last half a billion years, the ocean has mostly been full of oxygen and teeming with animal life. But earlier, before animals had evolved, oxygen was harder to come by. Now a new study led by researchers at the University of California, Riverside reveals that the ancient deep ocean was not only devoid of oxygen but also rich in iron, a key biological nutrient, for nearly a billion years longer than previously thought, right through a key evolutionary interval that culminated in the first rise of animals. Good morning, this is Dr. Will, CBMD, PhD. This is Thursday, September 8th, 2011, 5:26 a.m. Eastern Time. Quick note. Jumping genes' preferred targets may influence genome evolution. Science Daily, September 6, 2011. The human genes share several peculiarities with the DNA or just about every other plant and animal. Our genetic blueprint contains numerous entities known as transposons, or jumping genes, which have the ability to move from place to place on the chromosome within the cell. Good morning, this is Dr. Will, CBMD, PhD. This is Thursday, September 8th, 2011, 5:29 a.m. Eastern Time. Quick note. An astounding 50% of human DNA comprises both active transposon elements and the decaying remains of former transposons that were active thousands to millions of years ago, before becoming damaged and immobile. If all of this mobile and formerly mobile DNA were not mysterious enough, every time a plant, animal, or human cell prepares to divide, the chromosome regions richest in transposon-derived sequences, even elements long deceased, are among the last to duplicate. The reason for their delayed duplication, if there is one, has eluded biologists for more than 50 years. Good morning, this is Dr. Will, CBMD, PhD. This is Thursday, September 8th, 2011, 8:12 a.m. Eastern Time. Quick note. New material shows promise for trapping pollutants. Science Daily, September 6, 2011. Water softening techniques are very effective for removing minerals such as calcium and magnesium, which occur as positively charged ions in hard water. But many heavy metals and other inorganic pollutants form negatively charged ions in water, and existing water treatment processes to remove them are inefficient and expensive. Good morning, this is Dr. Will, CBMD, PhD. This is Thursday, September 8th, 2011, 12:11 a.m. Eastern Time. Quick note. Nanosensors made from DNA may light path to new cancer tests and drugs. Science Daily, September 7, 2011. Sensors made from custom DNA molecules could be used to personalize cancer treatments and monitor the quality of stem cells, according to an international team of researchers led by scientists at UC Santa Barbara and the University of Rome. The new nanosensors can quickly detect a broad class of proteins called transcription factors, which serve as the master control switches of life. The research is described in the article published in the Journal of American Chemical Society. N. Good morning, this is Dr. Will, CBMD, PhD. This is September 8, 2011, 4:40 a.m. Eastern Time. Quick note. Clouds don't cause climate change, study shows. Science Daily, September 6, 2011. Clouds only amplify climate change, says a Texas A&M University professor in a study that rebuts recent claims that clouds are actually the root cause of climate change. N. Good morning, this is Dr. Will, CBMD, PhD. It's Thursday, September 8, 2011, 12:14 a.m. Eastern Time. Quick note. New cellular surprise may help scientists better understand human mitochondrial diseases. Science Daily, September 7, 2011. A surprising new discovery by University of Colorado Boulder and the University of California Davis regarding the division of tiny power plants within cells, known as mitochondria, has implications for better understanding a wide variety of human diseases and conditions due to mitochondrial defects. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, September 8, 2011, 8:14 a.m. Eastern Time. Quick note. Chemists at the University of California, Santa Cruz have now developed a new type of material that can soak up negatively charged pollutants from water. The new material, which they call Slug 26, could be used to treat polluted water through an ion exchange process similar to water softening. And